about systemic infections. And systemic just comes from a designation meaning that this is throughout the body, okay? So affecting all systems. So a lot of these end up as becoming bloodborne pathogens and then exerting their effects on multiple organ systems. So we'll begin by talking about the anatomy and physiology of the circulatory system and the infectious process that can hit them. We'll describe symptoms that allow us to differentiate circulatory system infections from those caused by viruses, from those caused by bacteria and parasites, talk about prevention strategies and transmission routes, and talk about a treatment and prevention therapy for microbes that do systemic infection. And in this section, we're going to list the structural components of the cardiovascular and lymphatic system and their relationship to each other, compare and contrast systemic and pulmonary circulation, and then explain the relationship between the circulatory system and systemic infection. So, the job of the circulatory system is to serve as a transport mechanism to get oxygen and nutrients and hormones and immune components throughout the body. Okay? It also aids in the destruction of pathogens by the transport of white blood cells and other immune proteins such as complement. It regulates body temperature and it regulates body pH and has a role in our fluid balance. It's also our nutrient delivery and waste removal system for metabolically active tissue. The circulatory system you're most familiar with is the one that includes the blood vessels, and the heart, which is what's pictured here. Okay? It's split into two separate systems. The pulmonary circulation, which runs from the right side of the heart to the lungs back to the left side of the heart, and is extremely short and extremely low pressure, and whose job it is primarily to load oxygen into the blood while removing carbon dioxide, which is a waste gas and can't be allowed to accumulate in tissues, otherwise it'll produce toxic conditions that'll kill them. Okay? And then the left side of the heart, which pumps oxygenated blood to the entire body and returns that deoxygenated blood from the left side of the heart to the right side of the heart. Okay? That's a longer system, it's higher pressure, and it's greater in length. Okay? And that's why the most powerful chamber in the heart is the left ventricle, because it has to work against the greatest resistance. Okay? The blood vessels that move away from the heart are called arteries, and those that move towards the heart are called veins. Okay? The arteries tend to carry blood under higher pressure, while the veins tend to carry blood under lower pressure. As a result, the thickness and the muscularity of the wall of an artery is more extensive than that of a vein. Okay? Arteries become arterioles, which become eventually capillaries, which then become venules, which then become veins, and then those return back to the heart. Okay? The lymphatic system picks up some of the fluid that's left out at the capillary beds that isn't recovered by them and moves that fluid called lymph through structures called lymph nodes, which filter out pathogens and alert the immune system to the presence of the pathogen in the body, and then returns that filtered fluid back to the blood in the subclavian vein. Okay? In addition, the lymphatic system also transports dietary fat from the lymphatic system to the blood, and then from the blood eventually to the tissues, and specifically the liver. Okay? So that's the layout. All right? Notice that the distance between the heart and the lungs is very short, and that's to make the oxygenation of the blood more efficient. And the distance between the heart and the rest of the body is much more extensive. Okay? The pressures are much higher in the arterial than they are in the arterial system than they are in the veins, and that has to do with the fact that the veins are further from the heart and are receiving blood that's experienced a lot more resistance to flow. Okay, and as a result, to get blood back to the heart, especially from areas below the heart, we need the assistance of skeletal muscle and what we call the respiratory pump and the structures inside the blood vessels, the veins called valves, which ensure one-way flow. Okay. All right. The pump is the heart. Okay. The two lower chambers are called the ventricles. The two upper are called the atria. The atria receive blood, and the ventricles eject blood. That's why they're called the discharging chambers. Okay. The right ventricle discharges blood into the pulmonary trunk, which goes to the pulmonary arteries, which go to the lungs, while the left ventricle discharges blood into the aorta, which brings blood throughout the body, including to the heart itself. Okay? The lining of the heart is made up of three distinct tissues: the endocardium, the pericardium, the myocardium. The endocardium is simple squamous endothelial tissue, which forms a nice slick lining to the heart chambers and is continuous with the endothelial lining of the blood vessels. The pericardium is connective tissue that surrounds the outside of the heart and has both a visceral and a parietal component. The visceral component clings to the heart itself. The parietal component lines the chamber in which the heart sits. And in between those layers is pericardial fluid that reduces the friction of the heart as it beats. And then the myocardium is the middle layer, and it's made up of cardiac muscle tissue, which has the ability to contract on its own in a rhythmic fashion. That's a property called autorhythmicity. Okay? It's also set up to never go into tetany, okay, which is a state of sustained contraction that rapidly exhausts oxygen and nutrients. Okay? The one-way flow through the heart is ensured by a series of one-way valves. Okay? The one-way valve between the right atrium and the right ventricle is called the tricuspid valve. The one-way valve between the left atrium and ventricle is called the bicuspid valve. The one-way valve between the right ventricle and the pulmonary trunk is called the pulmonary semilunar valve. And the valve between the left ventricle and the aorta is called the aortic semilunar valve. Okay? Each of these valves only opens in one direction under normal circumstances. Right? Both of the atrioventricular valves only open into the ventricles, and both of the semilunar valves only open into the great vessels, the aorta and the pulmonary trunk. Okay? Otherwise, we wouldn't be effectively pumping blood throughout the body. Okay? An inflammation of any of these linings of the heart ends in itis, endocarditis, an inflammation of the endocardium, pericarditis, an inflammation of the pericardium, and myocarditis, an inflammation of the myocardium. Okay? The heart has a nerve supply, but the heart would beat even if there were no nerve supply to it because of the presence inside the heart of pacemaker cells, which are modified cardiomyocytes that generate an electrical depolarizing signal at a regular interval that precedes the contraction of first the upper and then the lower chambers of the heart. Okay? We call the contraction of the chambers systole. We call the relaxation of the chambers diastole. Okay? The autonomic nervous system innervates the heart. Both the parasympathetic and the sympathetic divisions of the autonomic nervous system can speed up the rate and force of contraction if we're talking about the sympathetic division, or bring it back down to resting level if we're talking about the parasympathetic division. Okay? Lymphadenopathy is a swelling of the lymph nodes. The lymph nodes are made up of macrophages and lymphocytes and reticulocytes and a network of extracellular matrix which contains structures called trabeculae. Okay, so these white blood cells sit on these trabeculae along with the um, the um, the cells that produce the extracellular matrix called fibroblasts. All right, and then fluid comes into these lymph nodes from the body. Okay, and percolates through them. Macrophages intercept invaders using phagocytosis. They swallow them up. They chop them into little pieces by fusing the phagosome with a lysosome, and then pieces of that pathogen are presented on the surface of the macrophage along with a protein called MHC2. Okay, and then those lymphocytes that recognize that display are the ones that are going to be selected to reproduce and then attack the pathogen. And those will include cytotoxic T cells and activated B cells. Okay? So lymphadenopathy is a swelling of the lymph nodes that generally results from the fact that it's fighting an infection. Lymphadenitis is an inflammation of the lymph nodes, which is painful usually. And lymphangitis is inflammation of the vessels that lead in to the lymph nodes. Okay? The lymph nodes in the body are positioned in areas that drain structures that have openings to the outside. Okay? There's a lot of lymph nodes in your neck. There's a lot of holes in your head which serve as ports of entry for pathogens. Okay? There's a lot of them in your axillary region because of the presence of the respiratory system and the presence of the breasts and the nipples, which have openings in them as well. Okay, and also the presence of the esophagus and the pharynx. Okay, which are open to the outside world. In addition, we have quite a few in the thoracic region, so in your chest, along the lungs, okay, and quite a few in the abdominal pelvic region where they drain the fluid that comes out of the digestive system, okay, and associated tissues, all right. And then we also have quite a few in the inguinal region because of the presence of, again, open systems such as the reproductive and the urinary system, as well as the back end of the digestive system. Okay? And so you always put your defenses where they're going to do the most good. That's why we locate the lymph nodes in those regions. And this is one of the ways that a doctor can determine that you're draining an infection, okay, is to feel for the lymph nodes in the different regions. And if you pick up swelling, the affected system is probably nearby, okay? The other job, of course, of the lymph nodes is to intercept and destroy cancer cells, okay? We'll talk about the pathogenic progress of systemic viral infection. We'll talk about the diagnosis of systemic viral infection to pathogenesis of specific viruses that make disease. And we'll talk about the signs and symptoms of systemic viral infection and the effect of the virus on the organ systems. And we'll list the treatment and prevention options for different systemic viral infections. Okay. So our first is mononucleosis, known as the kissing disease. The causative agent is the Epstein-Barr virus, which is a member of the herpes virus family. This is herpes virus 4. It's shed in saliva. Symptoms: fever, sore throat, enlarged lymph nodes, and fatigue. The virus infects and replicates in the oropharyngeal epithelium, so that would be the lining of the throat and the mouth, also in the tonsils and the salivary glands, and it produces B cells with an unusual appearance, as shown here. Okay, so you can See them here. Burkitt's lymphoma is cancer of the B lymphocytes. Tumors can form in the upper and lower jaw and the orbits of the eye. It grows rapidly, and the treatment is usually chemotherapy. Oftentimes, it's associated with Epstein-Barr. The reason for this is that viruses have the ability to integrate their genetic material into our genetic material. And when that happens, you can cause a change in the regulation of genes that affect the cell cycle, and that can turn a normal cell into a cancer cell. Okay, that's just one of the ways that this can happen. Okay, all leukemias, of which lymphoma is a part, are the result of mutations in the cells that generate the white blood cells. Okay, so you can have lymphomas, you can have acute or chronic leukemia of the cells that generate the granulocytes. Okay, so there's a large number of different types of leukemias. All right, so let's take a look at that now. Okay, cytomegalovirus is another member of the herpes virus family. Okay, this is herpes virus 5, and it's a double-stranded DNA virus that can integrate. Okay, and can, as a result, go into what we call latency, meaning it can sleep inside our own genetic material for the rest of our lives. Often people with it are asymptomatic and can shed the virus in body fluids like saliva and urine. It can affect cell-mediated immunity. Reinfections can occur with a second strain of cytomegalovirus and generate immunocompromised individuals that face a risk of reactivation. Symptoms include fever, pneumonia, hepatitis, which is inflammation of the liver, and encephalitis, which is an inflammation of the brain. Okay, we can also transmit from mother to child. Okay, we call that transplacental. The reason for this is it's tiny enough to cross the placenta. Okay, and this can result in birth defects, especially if this occurs within the first trimester. It's one of the reasons that we warn mothers, especially during the first trimester, to stay away from people who are sick. Okay? Dengue hemorrhagic fever is endemic in the tropics and causes intense joint and muscle pain, and uses mosquito as the vector. So the virus can persist in the gut of the mosquito, and when it takes a blood meal, it can introduce the virus into the victim. Ebola, very dangerous virus, has rapid onset of fever, muscle pain, and bleeding, and is transmitted through contact with body fluids. This literally can melt your body from the inside out. Fortunately, in a sense, because it's so deadly to its host, it usually doesn't end up going very far, right? It might reach an epidemic level, but people become acutely aware of the presence of Ebola as a result of the dramatic effect it has on the host, and at that point, usually steps are taken to control it that don't allow extensive spread. Okay, if you want to read a really good book on the Ebola virus, you should get The Hot Zone out of the library. Chikungua causes debilitating muscle pain and is prevalent near the Indian Ocean, Europe, and has also been found in the U.S. Here we're going to compare systemic signs and symptoms of bacterial circulatory infections. We'll generate a list of organ problems according to the pathogenesis of each bacterial pathogen we talk about. We'll relate the symptoms of systemic bacterial infection discussed with the progression of each disease, and summarize treatment and prevention plans for different infections that we talk about. Okay? So the whole idea behind the the immune system is to keep bacteria out of your blood. Okay, we don't want bacteria in the blood because then it can travel to any system in the body and cause a lot of organ damage. Okay, infection in the bloodstream is defined as sepsis. Bacteremia is bacteria in the bloodstream. Septicemia is a pathogen reproducing in the bloodstream, which can overcome our immunity. And septic shock is a drop in blood pressure due to severe sepsis. And the reason for this is the massive release of histamine by basophils that results in vasodilation and a huge drop in blood pressure. It is usually triggered by things like superantigens. Okay? Bubonic plague results from a bacteria that can move from the site of infection into the lymph nodes, forming buboes. It is generally not transmissible. The classic vector is the flea, and sort of by extension the rat. Okay. Septicemic plague is where the pathogen enters the blood and is not transmissible. And pneumonic plague is where the pathogen hits the lungs and is transmissible by aerosols and spreads quickly through populations. Okay? Bubonic plague you might also know as the Black Death. Okay? Virulence factors are features that bacteria have that cause them to produce disease. Okay? Examples include lipoproteins that can inhibit phagocytosis so they can avoid the macrophages of the immune system, an F1 protein capsule that blocks phagocytosis, biofilm formation where it attaches to a substrate and forms a multibacterial community. Okay? And this aids in transmission. Okay? And then type 3 secretion systems, which inject virulence proteins into the host. Okay? So you can see here an example of how bubonic can spread. Okay? You can see here that you've got the flea, and let's take the rat or some other type of rodent. All right? If you, as a human, have direct contact, you can pick up the infected flea from the rodent. It can bite you and take a blood meal, and then you can pick up the pathogen. Okay? So the idea here is that the rodent comes into contact with a flea. The rodent, after it's bitten by the infected flea, is now going to have infected blood, and then the infected flea from the rodent can bite the human, okay, and then even an uninfected flea can feed on the rodent and acquire the infection and bite the human, okay. Domestic rodents can do the same thing, okay. A flea is simply an insect that lives by taking blood meals off of its host, okay. The bubonic plague can progress to secondary plague pneumonia, which is the accumulation of fluid in the lungs as the tissues become damaged by the bacteria. And then you can have human-to-human transmission through things like aerosols. Okay? And so this is the Black Death. This is what killed a lot of people in the Middle Ages. And it's a pretty serious infection. Okay? Next, we'll talk a little bit about Lyme disease.
There are three possible ways for Lyme disease to be transmitted. Number 1, by the bite of an insect or tick that transfers blood. 2, in a pregnant woman, from the mother to the fetus via the placenta. And 3, sexually transmitted. Lyme also contains co-infections such as Babesia, Ehrlichia, Bartonella, and secondary infections such as Mycoplasma, Candida, and viruses as HHV6, cytomegalovirus, and Epstein-Barr. Lyme is characterized by a spirochete infection. The spirochete releases bacterial lipoproteins, that is a neurotoxin. The BLP neurotoxins are associated with symptoms like memory problems, burning and neurological pain, and numbness. The spirochete moves in a corkscrew-like motion as it burrows its way through the tissue and organs of the body. The spirochete contains antigens, and the antigens are like fingerprints, which identify this infection to the body's immune system. The dendritic cell is a key part of the immune system. One of the major functions of the dendritic cell is to process the antigen and present it to other parts of the immune system. This is why the dendritic cell is called the antigen-presenting cell. Once the spirochete enters the human body, it comes into contact with the dendritic cell of the immune system. The spirochete rubs or displaces the antigens onto the dendritic cell. Then the dendritic cell begins to process the antigen of Lyme disease for the other parts of the immune system to recognize it. The helper T cells come along and pick up the processed antigen off of the dendritic cell. Then the helper T cells proceed to pass the antigens along to another portion of the immune system called the killer T cells. The job of the killer T cell is to hunt down the spirochete infection and eliminate it by using the antigens to identify the Lyme disease. As the spirochete burrows into the body, it continues to spread its antigen onto the surface of healthy tissue and organs. The killer T cells follow as they seek out the antigen of the spirochete. In this process, the killer T cells begin to destroy healthy tissues and organs because they cannot differentiate between the antigen of the spirochete or the antigen of healthy tissue. As the killer T cells come into contact with the antigen located on the healthy tissue, we begin to see inflammation and destruction of the healthy tissue. This is clearly seen in autoimmune disease process, where the immune system cannot distinguish between self and non-self. As the spirochete is continuously moving, it's releasing the BLPs, the bacterial lipoprotein, which is impairing the immune system and causing inflammation, irritation, and damage to the peripheral and central nervous systems. The problem remains. The number of cases of Lyme disease reported to the CDC do not represent the total number of cases that exist. Lyme disease, its co-infections, and secondary infections may represent more than 300 different medical conditions, such as chronic fatigue, fibromyalgia, and other autoimmune diseases. The following diagram is designed to help explain the various scenarios of chronic Lyme disease treatment. There are three major focal points in the treatment of Lyme disease. One, the disease itself, which is represented by Lyme, its co-infections, and secondary infections. Two, the immune system, such as CD57 and other immune modulators. And three, the neurotoxins, or the bacterial lipoproteins in yellow, that usually take a longer time to clear up as the infection improves. As the immune system increases and the infection decreases over time, which is represented in the yellow line, we see the windows of symptoms improving. Also notice that it takes the bacterial lipoprotein neurotoxins longer to clear. This represents the ideal course of treatment. Unfortunately, this does not occur in the current methods of treatment. Now, let's review three common treatment scenarios. Number one, the patient is diagnosed with Lyme disease and then given 4 to 6 weeks of IV antibiotics. You'll notice it does nothing for the immune system except impair immune function. Slightly shifting the disease over 4 to 6 weeks, and then you'll see the relapse once the antibiotic treatment ends. You'll notice the antibiotics do nothing to address the co-infections, the secondary infections, or the neurotoxins related to the Lyme disease, and it does nothing to strengthen the immune system. In the second scenario, you'll see the patient is using IV antibiotics, dietary herbs, and supplements. The IV antibiotics bring the infection down a little bit, and the dietary herbs and supplements slightly increase the immune system. As in the first example, once the patient stops therapy, we see another relapse. The third scenario represents a patient using advanced immunotherapy. Notice the sharp increase and rise in the immune system. In addition to the IV antibiotics used to treat the infection, you'll see aggressive natural antiviral, antifungal treatments to fight the infection, and you'll notice a sharp decrease in the elimination of the infection. And finally, notice biodetoxification that is used to help the neurotoxin and the bacterial lipoproteins to be facilitated out of the body. This represents the Invita advantage in utilizing a comprehensive treatment plan to treat Lyme disease.
Okay, typhoid is caused by a salmonella bacterium called Salmonella enterica, and the serovar subtype is typhi. Okay, serovar just means that it reacts with an antibody to a surface protein that puts it in that group. Okay, and surface proteins usually are an indication of where it'll attach. Okay. It lives inside macrophages in the gastrointestinal tract. It infects a variety of organ systems, and the bacteria are continuously shed during infection and can be transmitted by a fecal-oral route. Okay, so contact with infected feces. Okay, and spreading that orally to an uninfected individual. Okay, so you might think, well, who's going out and touching infected feces? Well, it can be fecally contaminated water. Okay, it can be fecally contaminated water in the water table. All right, so it could be on things like the surfaces of vegetables or fruits. Okay, if you don't use purified water to do things like water your garden. Okay, or if you use a water source that comes into contact with infected feces, or if you swim in a public pool that has fecal contamination. Right? So those are just a few ways that you could pick it up. Okay? Next, we want to talk about Rocky Mountain spotted fever.
Good afternoon, and welcome to today's brief discussion concerning the disease Rocky Mountain spotted fever. What I hope to accomplish is to increase your basic understanding of the disease, how it is diagnosed, treated, and prevented. Rocky Mountain spotted fever, according to the Center for Disease Control, is the most severe tick-borne rickettsial illness in the United States. This disease is caused by the intracellular bacteria Rickettsia rickettsii, a delightfully redundant name. We'll call him Rick from now on. Rocky Mountain spotted fever is considered a seasonal illness, as around 90% of all cases are reported between the months of April and September each year. Contrary to its name, Rocky Mountain spotted fever is most common in North Carolina and Oklahoma, which compose 35% of yearly cases. The entire Rocky Mountain region, however, only contains 3% of cases. Between 800 and 1,200 cases are reported each year in the United States. This figure makes the previous statements even more apparent. You can clearly see the concentration of cases in a thick belt between Oklahoma and North Carolina, with Oklahoma and North Carolina carrying the most concentrated areas. Additional patterns that have shown up over the years is that the most common people to be infected are Caucasian males and children. In fact, 2/3 of reported cases of RMSF occur in children ranging from 5 to 9 years old. It's also been shown that people that spend extensive time around dogs are more likely to get the disease at least once, since they carry the most common vector for the disease. So by now you're probably wondering why it's called Rocky Mountain spotted fever in the first place if it's rare in the Rockies. And for that we're turning our attention to Howard Ricketts. Howard was a pathologist working for the University of Chicago when he became interested in RMSF after hearing about its discovery in 1896. He and a research team not only managed to isolate the responsible organism, but also its vector. This was in 1900. Unfortunately, Howard died a mere 10 years later in Mexico City after isolating the organism behind typhus, which is another rickettsial illness. He was killed by the organism he isolated. The organism behind RMSF is named in his honor. Now that we know a little bit more about our little friend's history, let's take a look at some of its general attributes. It is an obligate, intracellular, gram-negative bacteria that is non-motile and highly pleomorphic. It is able to take on the form of rods, cocci, and long strand-like threads based on its environment. It relies on eukaryotic cytoplasm for replication, and its home of choice is the endothelium of small to medium-sized blood vessels. In order to infect vertebrate hosts, it uses a vector in the form of a tick. The two most common ticks that act as vectors are the American dog tick and the Rocky Mountain wood tick. The life cycle of our tiny assailant is extremely convoluted, but it mostly looks something like this. Infected tick bites vertebrate host, an uninfected tick bites the infected host, the uninfected tick becomes infected. If the tick is male, it infects a female through mating. If the tick is female, it lays eggs containing infected larvae, which hatch into infected ticks, and the cycle starts over again. After entering the body through a tick bite, our friend Rick sets up shop. As stated earlier, he prefers small to medium-sized blood vessels if he can get them. Once inside a host cell, Rick replicates inside the cytoplasm or inside the nucleus, causing cellular damage. This leads to microscopic damage to the vessels, allowing tiny amounts of blood to escape. This escaping blood leads to the characteristic rash that is common in RMSF victims. This cellular damage can also directly damage organs. As far as signs and symptoms go, there are several. Initial symptoms include fever, nausea, vomiting, muscle pain, lack of appetite, severe headache. Later signs and symptoms include rash, abdominal pain, joint pain, and diarrhea. In other words, you really don't feel well. You're not a happy camper. There are also several ocular signs and symptoms, such as petechial conjunctivitis, anterior uveitis, retinal hemorrhages, cotton wool spots, retinal vascular engorgement, tortuosity, branch retinal arteriolar occlusion, and optic disc edema. As you can see, these signs are just as numerous as they are varied, and as a result, you will probably not be able to make a diagnosis directly from these ocular findings. Diagnosis comes chiefly from symptoms and history, as there is no fast lab test for diagnosing RMSF. Lab tests are usually done for confirmational purposes only. Physicians instead rely on the classic triad of fever, rash, and history of tick bite to determine if the treatment for RMSF is necessary. If the patient gets better, it was probably RMSF. If they want to confirm their diagnosis, they look for thrombocytopenia, hyponatremia, elevated liver enzyme levels, and a positive indirect immunofluorescence assay. The IFA is the reference standard for identifying RMSF. If the disease goes untreated, you're in a heap of trouble. RMSF has a 30% mortality rate if no action is taken, and a 71% hospitalization rate even if action is taken. RMSF can wreak havoc on your internal body systems, causing things such as gangrene, pulmonary hemorrhage, myeloencephalitis, and myocarditis. The good news is that RMSF is very treatable. Almost every case responds against doxycycline, the drug of choice. If doxycycline isn't an option, chloramphenicol is used instead. Of course, the best way to fight against the disease is to not catch it at all. The most effective yet most extreme method is to not go outside between April and September. A slightly more attractive option is simply to wear lightly colored clothing so the ticks show up better. You should also check yourself thoroughly after being outside in an area where you are likely to pick up ticks. The best course of action as far as clothes are concerned is to tuck your pants into your socks to prevent ticks from climbing up your legs. And although that sounds both hot and silly looking, consider this a possible alternative. Anyway, It's also good to spray your shoes and clothing with permethrin, which can last for days if you're out camping. Use spray containing DEET on your skin, but be careful. DEET only lasts for a few hours. Also be careful using DEET on children, as it is known to cause adverse effects in high amounts. Last but not least, if you see a tick on you, get it off as soon as possible. This brings us to a critical point on the proper removal of said tick. To properly remove a tick, you must grasp it as close to the skin as possible with fine-tipped tweezers. You must then pull up with even pressure. Avoid twisting, as this could cause the mouth to break off or the tick to regurgitate, increasing your chances of infection. After removing the tick, disinfect the area and wash your hands thoroughly. The CDC also recommends you place the tick in a plastic bag, date the bag, and then place the bag in your freezer in case you get sick. This would allow for a quicker and more accurate diagnosis of your illness. In conclusion, remember that prevention is a very powerful ally in defending yourself from RMSF. And if you have RMSF, the best course of action is to get it treated as soon as possible. If you would like to learn more about Rocky Mountain Spotted Fever, I highly recommend the Rocky Mountain Spotted Fever section on the Center for Disease Control website. Thank you for listening.
Okay, next we're going to name and define different manifestations of bacterial infections of the heart, compare and contrast different types of endocarditis, and talk about links between bacterial infection and atherosclerosis. So we talked a little bit earlier about these definitions, right? Pericarditis, infection of the sac that surrounds, anchors, and lubricates the heart. Myocarditis, inflammation of the middle wall of the heart where the cardiomyocytes that do the blood pumping occur. And endocarditis, an inflammation of the inner layer of the heart. The symptoms can be vague and intermittent. Examples of pathogenic agents include Staph mutans and Staph aureus. Okay, so again, the idea here is that they can colonize the endocardium, they can grow there, they can damage the tissue, and then they can produce inflammation. Okay. Here we're going to construct a list of systemic parasitic infections according to type and mode of transmission. We'll relate the different stages of systemic parasitic disease to the growth cycle of the infectious agent, and talk about the organ systems affected by each parasitic infection, and talk about a plan for prevention. Okay? So first up is malaria.
When a female Anopheles mosquito penetrates human skin to obtain a blood meal, it injects saliva mixed with an anticoagulant. If the mosquito is infected with Plasmodium, it will also inject elongated sporozoites, motile, spindle-shaped asexual cells, into the bloodstream of its victim. The sporozoites travel to the liver, where they enter liver cells and rapidly divide asexually. This asexual division, which is called schizogony, generates the next life cycle form, called merozoites. The released merozoites invade other liver cells and enter the host's bloodstream, where they invade erythrocytes. Once inside the erythrocyte, the merozoite begins to enlarge as a uninucleate cell termed a ring trophozoite. The trophozoite's nucleus then divides asexually to produce a schizont which contains several nuclei. The schizont then divides and produces mononucleated merozoites. The erythrocyte ruptures and releases toxins throughout the body of the host, bringing about the well-known cycle of fever and chills that is characteristic of malaria. Plasmodium enters a sexual phase when some merozoites in the erythrocytes develop into gametocytes, cells capable of producing both male and female gametes. Erythrocytes containing gametocytes do not rupture. Gametocytes are incapable of producing gametes within their human hosts, and do so only when they are extracted from an infected human host by a mosquito. Within the gut of the mosquito, the gametocytes form male and female gametes. The resultant diploid zygotes develop within the mosquito's intestinal walls and ultimately differentiate into oocysts. Within the oocysts, repeated mitotic divisions take place, producing large numbers of sporozoites. These sporozoites migrate to the salivary glands of the mosquito, and from there are injected by the mosquito into the bloodstream of a human, thus starting the life cycle of the parasite again.
Welcome to World Med School. My name is Lucille Bloomberg. I'm an infectious disease specialist in South Africa. In this micro lecture, I'm going to talk about the symptoms of malaria. Clinical presentation of malaria. Early diagnosis of malaria is critical to ensure early treatment and a successful outcome. Misdiagnosis and treatment delays are, however, common because malaria may present with very non-specific signs and symptoms. In many communities and amongst healthcare workers, there is frequently a low index of suspicion for malaria, and malaria is a rapidly progressive disease requiring urgent treatment. Symptoms and signs of malaria may present as early as 7 days after exposure, with a usual range of 10 to 20 days elapsing after being bitten by an infected mosquito. Longer incubation periods are noted in some cases of falciparum malaria, particularly in patients who've been taking some form of prophylaxis, which has not been effective, or they've not taken it in a compliant way, or they may be on other antibiotics, which may have some mild antiplasmodial effect. The onset of symptoms is typically sudden. Malaria symptoms in adults and in children differ. Most commonly, malaria presents as an influenza-like illness in adults, and the three commonest symptoms are fever, headache, and rigors. Typically, these occur in paroxysms with the onset of fever, then shivers, then sweats. In between the paroxysms, there may be a period of almost 48 hours during which the patient may feel relatively well, and there are no obvious signs and symptoms. However, as the malaria progresses, these periods may be reduced and the symptoms may be more continuous. Myalgia and fatigue are very common symptoms of malaria, but are very nonspecific. Loss of appetite may be noted, and in some patients, they may complain of nausea, vomiting, and diarrhea. Malaria symptoms in children are much more subtle. There is usually a history of fever. Lethargy is common. Parents may notice poor feeding. Vomiting and diarrhea may be noted. And particularly in children with complicated malaria, there may be a history of rapid breathing. The symptoms of severe malaria are particularly important to note because they do impact on the choice of treatment and the urgency with which it needs to be given. So any child or patient who is unable to drink, a young child who is not breastfeeding, repeated vomiting, patient being unable to sit or stand, prostration are particularly important to note. Shortness of breath is a very important symptom of complicated malaria, and cerebral malaria present with impaired consciousness, an agitated child, or even the presence of convulsions. Yellow eyes and dark urine may also be an indication of the presence of severe malaria. Certain groups of patients are at particularly high risk for the development of severe malaria. Any non-immune person who develops malaria and there is a delay in treatment may develop severe malaria. But pregnant women, those in the postpartum period, are particularly at risk. Infants, young children below 5 are an important group. Elderly patients, splenectomized patients, and those who are immunocompromised, particularly those with HIV/AIDS, are particularly at risk for the development of severe malaria. Malaria symptoms mimic many other infections, and vice versa. Many diseases that are common in the same areas where malaria occurs may look like malaria. Fever and rigors occur in malaria, but they also are common features of influenza. And if you look back on patients who died of malaria, influenza was often the first, the second, the third misdiagnosis. Dengue, typhoid, African tick bite fever, and East African trypanosomiasis are important considerations in patients who develop fever and rigors who live in areas where these diseases occur. Common diseases like urinary tract infections may present with fever and rigors. In fact, any common bacterial infection may present in a similar way, including septicemia. Viral hemorrhagic fevers are often confused in the early stages with the symptoms presenting that one would find in patients with malaria. Patients with a history of jaundice or yellow discoloration may initially be misdiagnosed as having a viral hepatitis, and yet may be malaria. And in fact, if a history of jaundice is present and malaria is considered, this would always indicate severe malaria. Change in mental state, convulsions, may be confused with meningitis or encephalitis or cerebral malaria. And patients with respiratory distress may be misdiagnosed as pneumonia, whereas in fact the respiratory distress may be a complication of malaria. So it is critically important that there is a high index of suspicion for malaria in anybody who has fever, a flu-like illness, plus a travel to a malaria area or residence in a malaria area, and in these patients it is malaria until disproved, irrespective of the time of year or visit to or residence in a low-risk area, or if prophylaxis has been taken.
Okay, now we don't want to talk about Bebasiosis and the insect vector for this, which is the tick. Okay, so it's similar to malaria, except that it's transmitted by ticks. Characteristic cell arrangement is what defines it. Usually forms a cross formation in the target cell. Okay, immunocompetent patients have flu-like illness, and patients with immunosuppression can have prolonged illness. Okay. Next, we want to talk about Chagas disease.
Definition. Chagas disease, also known as trypanosomiasis, is an infectious inflammatory disease caused by a parasite found in the feces of the triatomine or reduvid bug, which is very common in Latin America and the southern United States as well. Symptoms. Symptoms pretty much range from mild and acute to severe, and patients usually do not detect symptoms till the disease hits a long-lasting chronic stage. Acute phase. This usually lasts for weeks or months, almost symptom-free. Typical but mild symptoms that may show up include the following. Fever. Swelling around the infection, eyelids and glands. Fatigue. Body ache and headache. Rashes. Loss of appetite. Nausea and vomiting. Diarrhea. Enlarged liver or spleen. Acute symptoms usually go away on their own, but in some cases, if left untreated, they advance to the chronic phase. Chronic phase. This phase may never show up or may show up 10 to 20 years after the first infection. Typical symptoms in severe cases include the following. Irregular heartbeat. Congestive heart failure and sudden cardiac arrest. Abdominal pain or constipation due to enlarged colon. Difficulty in swallowing food due to enlarged esophagus. Risk factors. This disease, most often diagnosed in children, can literally infect anyone. Living in structures built with mud, adobe or thatch in rural impoverished areas of Latin America where the triatomine bugs are rampant. Traveling to a high-risk area of Chagas disease. Blood transfusion or organ transplant from someone with Chagas infection. Diagnosis. The doctor will ask for the following. A physical examination to check signs and symptoms of the disease or any risk to the disease. blood tests to confirm the presence of the disease in the bloodstream. Additional tests like ECG, chest and abdominal X-ray, echocardiogram and upper endoscopy to check the stage of the disease and the resultant complications. Treatment. Prevention of infection is always ideal. The first line of treatment is to kill the parasite in acute infection and then go about managing signs and symptoms in later stages. If left untreated, it can cause severe heart and digestive problems. Treatment for heart-related complications include medications, pacemaker or any support device to regulate the rhythm of the heart, surgery or a transplant. Treatment for digestive-related complications include a diet change, medications, corticosteroids and surgery in extreme cases. Medications during the acute phase may benefit the patient, though not so much in the chronic stage. The medications during the chronic stage don't cure disease but helps slow the progression of the disease and complications that come with it.
Toxoplasma gondii is a protozoan parasite of the phylum Apicomplexa. It is distributed worldwide and can infect all warm-blooded vertebrates, where it develops as an obligatory intracellular parasite. Its final host is the cat. With feces, the cat sheds environmentally resistant oocysts. These sporulate outside the host to infectious stages. For the development, called sporulation, humidity, temperatures of about 20 degrees Celsius and sufficient oxygenation are required. Inside the oocyst, at first, 2 sporoblasts are developing. Within the next 2 days, they become sporocysts. They are now surrounded by a wall and each contain 4 sporozoites. Having entered the intermediate host, excistation occurs in the lumen of the intestine. Under the action of intestinal enzymes and bile salts, first the oocyst is opened. Here we see one isolated sporocyst still wrapped in the ruptured oocyst wall. With digestion of the sporocyst envelope, the parasites are eventually set free. Once more, the process of excistation. The parasites are termed sporozoites and are the organisms infectious to all intermediate hosts. Sporozoites are formed as elongated cells of 7 by 2 micrometers. They show active gliding motility where the parasites are gliding on a glass slide. All single zoites, sporozoites, and, as we shall see later on, tachyzoites are similarly structured. The zoite contains a basally located nucleus and other organelles, just like any other eukaryotic cell. A typical feature is the apical complex. It is composed of a conoid and 3 types of secretory organelles. Small vesicles, the micronemes, are believed to play a role in host cell recognition. The rhopteres are elongated club-shaped organelles, 8 to 10 of which are found in each zoite. The third kind of secretory organelles are the dense granules, which will play a role after the invasion of the host cell. Infection of intermediate hosts occurs from oral uptake of oocysts containing sporozoites. Raw or undercooked meat containing tissue cysts loaded with invasive organisms named bradyzoites is a further source of infection. Once in the host, the parasite crosses the intestinal cells and starts multiplying in various cell types such as macrophages, fibroblasts and neurons. At this stage, which is the acute phase of the disease named toxoplasmosis, a transplacental transmission is possible. This can be fatal to the fetus. The invasion of host cells can also be observed in vitro in tissue cultures. The invasion of host cells is based on active motility. Parasites glide on glass slides, on the cell surface, and even within the host cells. They always glide on a substrate. The mechanism is not yet fully understood, but involves an actomyosin motor located beneath the parasite membrane. The occurrence during the invasion will now be shown in detail. Having reached the host cell by gliding, the apical contact triggers signals that lead to the exocytosis of micronemes. Their contents create a stable bond between the parasite and host cell. The junction is then moved posteriorly. At the parasite apex, a vacuole forms, which develops further with the apical discharge of lytic substances from one or two of the rhopteres. The vacuole membrane remains continuous with the host cell membrane throughout the process, but no host cell protein diffuses into the vacuole membrane. The vacuole membrane is a composite between host cell membrane lipids and rhoptere contents. After internalization, the dense granules move to distinct exocytosis sites to be discharged into the vacuolar space. The released material accumulates and is gradually associated with contorted tubular structures termed the vacuolar network. Dense granule and rhoptere proteins associated with the parasitophorous vacuole membrane are believed to participate in the organization of transmembrane pores. These may contribute to the nutrition of the parasite and leave free access to molecules up to 1500 Daltons. Finally, host cell endoplasmic reticulum and mitochondria accumulate around the parasitophorous vacuole. Host cell invasion observed here in tissue culture only takes a few seconds. Apical contact is followed by smooth gliding inside the cell. The constriction at the site of entry can be seen during the whole process. Electron microscopy would reveal the moving junction here. After invasion, the parasite begins with its intracellular development. This is shown in a tissue culture of human fibroblasts. This is a close-up of an early vacuole. A major feature of the vacuole is that it never fuses with any other compartment of the host cell endomembrane system. Within the vacuole, the parasite begins to multiply. Multiplication is by repetitive binary fission and is referred to as endodiogeny. It is a very complex process, where, after nuclear division, two daughter parasites form inside the parent organism. They will eventually emerge from the parent, which will gradually disappear, being progressively incorporated into the daughter zooites. Repetitive multiplications produce further parasites. They are called tachyzooites. Each division takes approximately 6 to 12 hours depending on the strain. The fastest growing parasite is usually the most pathogenic in vivo. The vacuole grows in size and will eventually occupy most of the host cell volume. It finally reaches a stage when the host cell cannot physically cope with further parasite development and ruptures. The egress in this case has been artificially triggered by calcium ionophore. Free tachyzooites will then disseminate throughout the entire host and infect new cells. Active invasion also occurs in macrophages, which are a most favored host cell in vivo. In this case, the process is identical to what was shown before in fibroblasts. A moving junction is formed and invasion is completed within a few seconds, just as in any other cell type. When the host starts generating an immune response, tachyzoites are efficiently eliminated by macrophages. This mechanism of host defense, called phagocytosis, can be shown in cell culture. Phagocytosis is not oriented apically as with invasion. The parasite is surrounded by the cytoplasm of the macrophage and incorporated. This process lasts more than 1 minute. After phagocytosis, the parasite is killed and digested. Escaping parasites are able to maintain a chronic infection. These parasites developing in cysts are termed bradyzoites and grow mainly in muscular tissue. The cysts can persist for years. They are usually controlled by the host's immune response and remain quiescent within the tissue without causing any harm to the host. However, when the host suffers immunodeficiency, such as in AIDS or upon organ transplantation, the infection can be reactivated. Bradyzoites can be released and switch back to the tachyzoite stage. This acute phase can be fatal in humans. Cysts are the main source of transmission to final or intermediate hosts. A cyst is surrounded by a thin wall which is made by the parasite as a deposit under the membrane of the parasitophorous vacuole in the host cell. Cysts can be isolated and aggregated by gradient centrifugation. The host cell is destroyed in this process. Bradyzoite release can be triggered by proteolytic enzymes such as trypsin, just as occurs in the gut of a carnivore after eating infected tissue. First, the bradyzoites invade enterocytes, later other tissues. There they multiply as tachyzoites, resuming the intermediate host cycle. Ingestion in the intestine of a cat leads to the invasion of enterocytes, where the bradyzoites undergo a typical coccidian cycle. In the epithelial cells of the duodenum, multiplication or schizogony takes place. The resulting merozoites invade neighboring cells. They develop first into macro and microgametocytes, then into macro and microgametes. This is known as gametogenesis. By fusion of the flagellated microgametes with the macrogametes, an oocyst is produced. Oocysts are excreted unsporulated. That means that they contain a single cell or zygote. The thick oocyst wall protects them against external influences and is extremely resistant to disinfection. It can persist infectiously for several months. Inside the oocyst wall, sporulation takes place. It lasts for about 30 hours and is shown here in time-lapse. With this development phase, the life cycle of Toxoplasma gondii is completed. After oral uptake of these infectious stages, the cycle can begin again.
Okay, leishmaniasis has both a cutaneous and a visceral form. Cutaneous form is caused by Leishmania tropica and Leishmania brasiliensis, while the visceral form is Leishmania donovani. Patients die usually within months due to hemorrhage, anemia, and bacterial pneumonia, and the treatment is antiparasitic drugs such as ivermectin. Okay? How do we transmit? Well, larvae are transmitted to humans through the insect bite. The adult worms develop and multiply in the human lymphatic system, and they nest there and mate and produce millions of microfilariae. These are worms which then enter the blood. Lymphatic blockage results from the presence of these worms, and inflammation causes accumulation of lymph and swelling in the affected areas. So you usually see these where there's lots of lymph nodes, which would be areas like the inguinal, okay, the popliteal, the axillary, okay. So you could see some examples here, right? This is an infection with Bancrofty, and then here's an example of elephantiasis. Okay, this interferes with circulation through the region and can result in tissue damage. Okay, then the infected individual has microfilariae ingested by a second insect, which is the intermediate host that takes a blood meal. The larvae progressively develop from L1 to L3 in the mosquito, okay? And then the infected mosquito bites an uninfected individual, and we repeat the cycle, okay? All right. That brings us to the end of this chapter. I will see everybody in the next podcast, and I thank you for listening.